Hey, thanks everyone. So earlier this year, Ellery and Sunny contacted me because they said they'd heard a rumor that I could say something about nitrate in braided rivers. So indeed, I've done a lot of work on nitrogen in rivers, and I've recently started to do more work looking explicitly at nitrogen in braided rivers. Um, so with this talk, I really want to make, I guess, a pitch for all of you that this is a worthwhile thing to think about from the scientific point of view, how nitrogen moves through these distinct rivers. And also that perhaps it's yeah, really exciting and something to consider for maybe more practical purposes. Yeah, it's something complicated and exciting. Hopefully, that's the goal of this talk. So that's what I hope you leave thinking of. So before I get into the specifics of braided rivers, I want to just take a step back and think about why we care about nitrogen in rivers globally, just full stop. So nitrogen is, of course, totally fundamental to life. It's in all of our DNA. It's in everything's DNA. And within that context, rivers play a really unique role in moving nitrogen from the land into the water. So how much nitrogen is in a river? It will be important for the function and life within that river, how stuff grows in it and what that ecosystem looks like. And how much nitrogen moves through the river will also be really important for what happens downstream. But of course, a lot of what we worry about and talk about and a lot of our science around nitrogen is really motivated by concern with how much extra reactive nitrogen, so the kinds of nitrogen that can be kind of feed life, how much extra of that us humans have made over the past 100 years or so. So over the past 100 years, we've increased the amount of reactive nitrogen moving through the world by 200%. And just to put that in context, like the amount of CO2 flux we've increased through fossil fuel burning is just 2%. So the human alteration to this kind of global nitrogen cycle is astronomical. And because nitrogen is so important for life and so mobile, it's really important to try to figure out what that's actually doing, both maybe within the river, that those kind of concentrations within the river will affect the ecosystem there, and also what it might be doing in terms of transport and linking these different ecosystems together. So what this means in practice is that, a lot of councils and water quality monitoring happens, and you kind of go out and collect a sample of water, and you see how much of something like nitrate is there. And you'll also often see water quality monitoring data that will have this total nitrogen or TN value as well. But I guess, yeah, before I go further into the talk, I think it's important to have a little bit of perspective, but not too much, like don't get too stressed out about it, of all the other kinds of nitrogen that are out there. Nitrogen is really, really biologically reactive, really biologically important. And there are a whole bunch of different forms of it all the time in the environment. And because it's so important for life, those forms are always being recycled everywhere you go. But what we really think about, and it's also feeding life into organic forms. But why we measure things like nitrate is because nitrate is physically, it moves really easily from kind of land into water. So it's a really good indicator usually of, say, if there's been kind of a human pollution effect, which is mostly what water quality monitoring is concerned about. And there might be some more kind of toxicity effects with that. But yeah, mostly there's more toxic forms of nitrogen. It's just that it's very mobile. There's generally a fair bit of it if humans have done something. So it's kind of a useful thing to measure if that's your target goal. You also see a lot of total nitrogen. Total nitrogen is everything that's there, excluding these kind of gas forms, which are kind of only interested in, like, important if you're interested in greenhouse gases like N2O, and N2 doesn't really do anything. So yeah, total nitrogen is something that's measured mostly when people just want to make sure that they get a number in terms of just making your data set. I would say, yeah, the chemical methods that you use to measure total nitrogen, if you want to think about that part of it, are pretty crappy. So those numbers are not very precise, but you're always guaranteed to measure something. So it's great if you want to make sure your data always has some number in it. But anyways, from that point of view, we've New Zealand and Canterbury in particular, there's a lot of concerns about how much extra nitrate in particular is in a lot of the waterways. So there's been a lot of monitoring work. There is a lot of data out there collected by people like ECAN. And a lot of the kind of approach of this work is really to think about how are these concentrations stacking up to some limit. And these limits can have a lot of arguments about what the limit should be, and there have been a lot of arguments about what those limits should be. I think that top line is maybe the current limit, and people maybe want to lower it to the other lower one, or maybe even lower. But whatever, those limits are all kind of thinking about, a toxicity effect. So how much nitrate concentration could be in the water before it starts being bad for human health, or being bad for fish, or some other cute thing that's living in the river. And from that point of view, now kind of going back to our braided rivers, you can say like, okay, well, here's 8 braided rivers, all with quite different kind of locations and hydrology across Canterbury, and they're all way below those limits. So we don't need to stress about them. We should really focus our stress on like groundwater and small streams, where indeed there are definitely streams around Canterbury that are way above that top limit. And you can talk to a lot of people about this, and they'll say, oh, yeah, you don't need to worry about nitrogen in braided rivers, because braided rivers are really big. They have a lot of water in them, so any nitrogen that ends up in them will just be diluted. It's like basic math. Small amount, the same amount of nitrate can go into that river. It'll be diluted, so we don't need to worry about it. And that kind of up a little bit with this data I've kind of collated from ECAN. So the ones on the left with the lighter colors are the kind of smaller hill-fed streams. So the kind of mean annual discharge, those numbers there, and they're about an order of magnitude lower than the rivers on the right, which are the kind of alpine mountain-fed rivers. They have a lot of water moving through them at all times. So it would take a lot of nitrate entering those rivers to have a measurable difference in the concentration. So that's kind of fine if your only concern is like whether the amount of nitrate will kill a fish or yourself. But I don't think, just looking at that, we haven't really explained the full story of perhaps like why the Orari has a lot more nitrate in it than the smaller Conway River. So there's a lot more complexity in that data, especially if you kind of remove the kind of upper limits and start thinking about what might actually be driving these differences, rather than just like, is it going to kill us or not? So one pretty simple thing that I've done in a lot of other rivers I've worked in to start to kind of get a more sensitive way of measuring not just is nitrate dangerous, but what's actually happening to the river as an ecosystem and how much nitrogen is moving through it, is to take this concentration data and simply kind of remove the hydrology part of the equation by multiplying it by how much water is going through there. So that kind of accounts for that discharge effect, and you can really see whether the amount of nitrogen moving through the river has increased or decreased over time, regardless of what the hydrology is. So here, sorry, I haven't labeled this. That's the Selwyn Waikiri at the top, and then the Rakaia, and then the Waimak, which for some reason I've missed a bit of data for. So you see definitely, the Rakaia is a much bigger river. There's kind of massive amount of water moving through it. And the Selwyn does have somewhat more nitrate in the water column. But when you multiply those together, you start to see a little bit more detail and more trends in those data. And I mean, I have put trend lines in there that do show them all increasing, but that was quite a quick calculation, so don't know if that's really true. But what you start to see is that these big rivers with very low nitrate concentrations are moving a significant amount of nitrogen from land to the coast. So even though it's a small number in terms of the amount that might be in a liter of water from one of those rivers, it could have a significant effect on the downstream ecosystem, and it has a significant effect on how those kind of the whole ecosystem fits together. There's a problem with that approach, and that's when we have a braided river. You have to think about where that data comes from. And, in practice, you drive along SH1, you get down and you grab a sample from the biggest, fastest flowing part of the river. And that's kind of where a flow gauge might be that people are trying to model the total discharge of these rivers from. But as Holly has talked about really well in her previous talk, braided rivers are really complicated environments with all different channels that are intermingling and have different amounts of water in them, and that water is flowing at different speeds. We also know that not only is water moving down them and across braided rivers, there's also constant exchange with that kind of shallow braid plain aquifer underneath the river. That means that you just have, constant mixing in kind of 4 directions maybe. So is that nitrate concentration that we've measured at one point really representative of what's happening across this entire ecosystem? And there's reason to think not. When we go back to this giant like whirl of the nitrogen cycle, all of those reactions are driven by biology, and like not biology in the cute sense, but biology in terms of microbes and fungi and stuff like that. And we know that how quickly those reactions happen basically depends on things like temperature. Warmer things happen faster when it's warmer. How fast the nitrogen moves, so how fast the water's flowing, how much time there is for a reaction to occur, and then how much kind of biomass or carbon is there, how much life is there already to kind of absorb that amount of nitrogen. And as Holly showed, across that braid plain, there's a huge variety of all 3 of those factors. So you'd expect that maybe how efficiently the braided river kind of exports versus holds on to nitrogen varies across that braid plain. So that's essentially what I'm setting out to look at. The idea being that maybe in some zones of a braided river, when you have that really complex structure, you have zones of growth that are consuming nitrogen, and then maybe you have zones of export where nitrogen is moving more efficiently through the river to the coast. So I've just started working on this, and mostly Holly has done this work for me. So I started to do a bit of a pilot study at her Cass River field location to start to get a handle on how much variety there is in these kind of biological reactions and productivity of these different zones of the braid plain. So she set up stage height loggers at 5 different locations. So the darker colors are the main braid, so faster flowing and deeper. Then we had 2 side braids, so a bit shallower, and then 1 spring, where we put loggers in to try to get a sense of the flow. And then I also went in and put these dissolved oxygen loggers that will measure oxygen every 10 minutes and as well as light and temperature. So with that combination of data, just to highlight the flow data isn't that great, but with that combination of dissolved oxygen, light, and temperature, we can start to get a sense of kind of how productive the biology is just as a composite in these different zones. So just to the big struggle that we've had that I'm also keen to talk to anyone about is just that the water moves way too much across these braid plains. So looking at the spring, we've had a logger in there for 4 or 5 months. It's great. It has a very consistent water height. The spring is, staying the same all year round, which makes sense. The side braids, both of them, we put the loggers in, and within about 5 days, there was no water left in either of those channels. It had migrated across the braid plain, so there's not much data from those. And similar problems with trying to get the main stem. The biggest part of the channel, we're able to pretty consistently have water in it. But the other point that we thought was like, surefire, definitely there's a lot of water and it's going to stay there. Again, after about 4 or 5 days, that channel migrated somewhere else. So the data that we actually have on dissolved oxygen is pretty limited because if there's no water, we can't measure dissolved oxygen. Looking at the bit of data we have been able to collect, or Holly's been able to collect, this is the dissolved oxygen saturation in the spring. So the point of these graphs is just that a bigger amplitude of night to day means that there's kind of more life in that spring. So we actually see really big fluctuations between night and day. So the low points are at night and the high points are during the day. So that was exciting that there's definitely something happening. And comparing it to that bit of data that we got from the main part of the braid, there's also something happening, like much smaller fluctuations. But I was really surprised that we actually do see this diurnal cycle, implying that, perhaps there is some activity within this kind of fast flowing and at that point fairly turbid bit of the river. So just quickly, with that data, with those night-day cycles, you can start to estimate like total ecosystem respiration, it's called. So that kind of total productivity, how much oxygen or carbon is being consumed and produced from these different parts of the river. So for those fast flowing turbid bits of the river, we actually do see some amount of respiration happening. As I said, does seem to be activity within those components, and the faster flowing main braid was less active than the side braid, which makes sense. But then those are all blown away when we add the spring site into it. So a much more negative number means it's consuming a lot more oxygen, it's respiring a lot more. So that slow, like relatively steady spring was super active. And also with that spring, because there was water there all the time, we're able to get some seasonal data and see that when we started kind of in the summer compared to the winter, there's been a significant decrease in how much it respires, which again just makes sense. It's colder, there's less daylight hours, it should be less active. But this is really like a proof of concept, starting to say that yes, these changing flows across the braid plane are changing biology, not in the cute sense, but in the sense of how elements are cycled through the river, how the microbes are processing things is a composite. So it's really cool that that's been verified despite the challenges of trying to get data across these constantly shifting rivers. But now the challenge is to say, well, first to connect it to actual measurements of nitrogen, but then also to say, okay, does what happens in that little spring actually matter to the river as a whole? So kind of going back to what I said at the beginning, that you can kind of take a concentration and multiply it by the discharge to get a sense of how much flux moves through a system. Does this kind of really reactive spring that has a very little amount of water in it, so that's the estimated discharge just based on it, has like 10 centimeters of water in it, not going very fast. Does that actually matter to what happens across the entire river as a whole when you compare it to those really fast flowing channels that are constantly exporting stuff downstream and also constantly exporting stuff to the air? So turbulent flow will also move more stuff into the air than those slower flowing springs. So that's what I had to talk to you guys about today. So I guess as next steps, as I said, was putting more of these loggers out in more systems. I'm particularly interested in setting up sites moving across the Braid Plain in any of these 8 rivers. So I'm keen to talk to anyone that knows of good sites and good access on any of those systems, and in particular, kind of matching up the data sets I'm collecting, which are really affected by the biology there, like is there a tern colony, but also the hydrology. So I'm keen to talk to anyone that's doing work on any of these rivers as well. Thank you very much. Yeah, exactly. And I think that the fact that, like I said, that these rivers are so big, it means that you can really do a lot of things like that, and there's no kind of measurable effect when you just look at the concentration change, right? Even if you're pumping quite a lot of really concentrated nitrate in, that probably does have a local effect. But in terms of, okay, you just grabbed a sample somewhere else, it would take so much to change that. It's like putting something in the ocean, right? Yeah.